Welcome back. Now that we've taken a look at the powering of the muscle, now we can take a look at the contraction of the muscle a little bit closer as it pertains to twitches in the muscle, or what we call a muscle twitch, and how that eventually ends up being our full contraction of the muscle itself. And so this is where we start to talk about summation in muscle. And so with neurons in the nervous system, we talked about summation and in the processes that were happening there in terms of a temporal and a spatial summation as well. And here we have a similar kind of a concept within the muscles also. And so spatial summation is essentially where within a muscle we have, remember, we have motor units. And so these motor units are the individual neuron and all of the muscle cells that it actually innervates. And so if we have spatial summation, we're going to have multiple motor neurons or multiple motor units all within the muscle contracting essentially all at the same time. And so with this, we have the more motor neurons that we stimulate within the muscle, the more muscle fibers that are going to get contracting. This equals a stronger contraction. So this is essentially kind of giving a larger stimulus all at the same time to all of the motor neurons within the muscle itself. Versus temporal summation, this is kind of over time. So this is, we're sending a signal to the muscle more frequently. And so we're getting a contraction of the muscle that is kind of increasing and increasing and increasing over time rather than having it all at one time. And so the more frequent the stimulation, and so in a sense, this one is we've got stimulation coming and going, and then all of a sudden it becomes faster. That number of stimulations equals a more frequent contraction, a contraction of the whole muscle at a greater frequency. And so this starts to become summative or additive within the muscle itself. So we begin with the concept of just a muscle twitch. And so a single muscle twitch or a muscle contraction is what we essentially start out with. And so here we have our point in time where we stimulate the muscle. And this is essentially within a experimental model within the lab kind of thing. And after we stimulate the muscle, there's a period of time where essentially nothing happens. And this is our latent period. This is the period of time where kind of the slack needs to be taken out of the muscle. The muscle itself is not generating any force at this time. And especially because it's not a muscle that's within the body per se, it's in a rig, that tension needs to be taken out of the muscle. And then we hit the contraction phase. And so the contraction phase is now that period of time where the amount of force is increasing. So the amount of tension that the muscle is generating is going up. And based upon the strength of the original stimulus is going to basically dictate how far up that goes. And so that's going to determine how much of a contraction we get. And then we have relaxation phase. And so that relaxation phase is now where essentially during the contraction phase, calcium ions were coming in to the cytoplasm. And during that relaxation phase, calcium ions are going out of the cytoplasm. And so we're pumping them back out. They're coming off of the troponin, they're leaving the space, and we're now getting to the point where we have the ability to relax the muscle and stop that contraction that is occurring. If we were to hit it again with another stimulus, say over in here, then we would go through the same thing, and we go through our latent period, and then we go through our contraction phase and relaxation phase. If we were to start that stimulus back over here, and we were already in the relaxation phase, and then we hit it again, now we're going to get another stronger contraction. And so that's the basis behind this process of increasing the amount of stimulus to an individual muscle itself. And so here we're looking at the triceps muscle. And for spatial summation, we've got, for example, 3 motor units. Each one of those motor units, we can see that they control a different set of cells. They control a different amount within the muscle itself that's there. And depending upon the amount of stimulus that we want to give, as we tell that whole muscle to contract. We can do it spatially and tell the entire muscle to contract at the same time. We can tell individual motor units. Okay, motor unit 1 fire off, motor unit 3 fire off, then motor unit 2 fire off, and we can kind of vary the amount that is in there as well. And so we can change that overall degree of stimulation that we're getting to the muscle at any given time. In the lab, we measure this using a process to be able to look at the amount of contraction that is happening with the muscle. And so we have a grip force device, and you sit there and you squeeze the device itself. And so in the lab, you would be squeezing on a grip force indicator. That then goes into the machine, and then we print out essentially the subsequent information that we get from there, which in this case, we're seeing spatial summation. So we're increasing the amount of force that is occurring because we're increasing the amount of motor units that are involved. And so in the lab, we asked the students to do a contraction that is 25% of their full strength, 50%, 75%, and then give everything that they can in terms of grip. And you can see that as they go through this process, the amount of force that is being generated keeps going up and up and up as we go through the different processes themselves. We're involving more and more and more of the muscle in order to get to that point, which means that we're having to spatially summate by increasing the amount of motor units that are involved. And so this one maybe has, in a sense, is going to have 25% of those motor units. If we had a 4 motor unit muscle, this one would have 1 motor unit. Let's go MU. This one would involve 2 motor units, 3 motor units, and then 4 motor units. And we would be up to the maximum for that particular muscle itself. And this allows us to see that as we change the amount of force that's being generated, the amount of contraction that we're giving, we're able to change the amount of force that is there. And as we're doing that, if we were to ask the person to say, stay at 25% and maintain the amount of tension that is there at roughly 25% of their ability, then we would see that they're switching from 1 motor unit to the next motor unit to the next motor unit, and it's varying off and on and off and on. And so after the first round goes, then we come back to number 1, number 2, and number 3 once again. And we're able to limit the amount of fatigue that is there. And so we can sustain that 25% contraction, 50%, even 75% contraction for a longer period of time because we're basically using different parts of the muscle at any given time. And that allows, as number 3 is contracting here, both number 1 and number 2 have been relaxing. Now number 1 is going to start to contract and number 3 gets to relax. And we keep going through this process over and over and over again as we continue to use that muscle, and fatigue then becomes kind of pushed to the back, where we're able to sustain that contraction over a longer period of time. So with temporal summation, we have the situation where when we look at an actual action potential and we look at the process itself for the muscle, and we have our contraction and our relaxation phases here, the entire contraction itself for a given muscle, this could be in the neighborhood of, over time, 100 milliseconds. And so we've got a fair amount of time. But the actual action potential, the stimulus that happened back over here, this only happens in 1 to 2 milliseconds. So in theory, if we had a 1 millisecond action potential, we could stimulate this muscle 100 times in the course of that particular contraction. And so we have the potential where that stimulus can be added on and added on and added on over that period of time. And so if a second stimulus happens right in here, say, now that action potential again causes more release of calcium. We already, in a sense, still had, so the calcium in the contraction was going in, and the calcium during the relaxation was going out, and we still have some of that calcium still inside the cell. inside the cytoplasm. So if we release more calcium at this point, instead of the curve going downward, the curve is going to come back up. And then it's going to come back down. And then if we hit it again, then it's going to come back up again. And so this temporal summation is going to lead us to a slightly different look in terms of a graph in this process. So we've got more calcium now in the cell, more binding sites are open, more cross bridges can be formed, and we get that stronger contraction. And so that's why each time here it goes up further as we go along through the process itself. And so it goes up and up and up over time. The same thing here is going to happen in that we're going to change the motor units that are there, that are involved, in order to prevent fatigue. And so we're still going to be changing from motor unit 1 to 2 to 3 to 4 and so on, and alternating as much as possible, thereby avoiding the fatigue that may be starting to occur. And so here we have a wave summation. And so we have our first stimulus here, and we get the first part of our contraction. Now, before it has time to completely relax, we hit it again, and we've got now calcium in even greater amounts. That opens up more binding sites. That allows for a stronger contraction. Now it starts to relax again, and we hit it again, and we get an even stronger contraction. This is going to increase and increase and increase over time until we reach potentially maximum contraction. And then no matter how much more we keep on stimulating it, it can only get to that maximum point. And so if this is, say, 5 kilograms that muscle can move, that's all that it's going to be able to do until it hits to that maximum point. And so it can kind of increase and increase over time, but it has a limited, or it's limited by the fact that it can only go up to the maximum force that particular cell is able to generate, or that particular muscle is able to generate. This can occur if we start to now decrease the amount of time in between the relaxations. As we kind of complete this process itself here, we get up to the point where the muscle is contracting and we're stimulating it, and we're maybe at like 80% of contraction on this one. There's still some relaxation. The stimuli are not coming fast enough. We've increased it a little bit and we've gotten up there, but there's still some relaxation occurring. So this is an incomplete tetany. And so the tetanus has not been complete. We're not reaching that point where it just flatlines at the top and it stays contracted at maximum contraction. But they are coming very rapid. This is coming faster and faster and faster within the muscle cell itself. And so they're starting to twitch even faster. Complete tetanus is where the frequency of those stimuli are now coming essentially too quick. And so we go very quickly up to maximum contraction of the muscle. Calcium is essentially staying inside the sarcoplasm. It's staying inside there, and calcium levels are at their greatest inside the cytoplasm. This results in a peak as far as that muscle is concerned. And again, if that's like 5 kilograms, then that's the most that it's going to be able to do. No matter how much we increase the rate of stimuli, if we put stimuli even in between these and increase the frequency that's there, it's still only going to be able to generate that 5 kilograms of force. But it's going to be a sustained force. It's going to stay at that 5 kilograms until we completely fatigue the muscle out or we stop the stimuli. So just kind of a recap of where we were at there. And so here we have our single twitch. It goes through its contraction and relaxation completely. Our wave summation can happen where we have 2 stimuli in one sequence. We can continue to increase that and get unfused tetanus. And if they're coming enough, instead of going all the way back down, they're just going to kind of stay up here and still see that twitching occurring within the muscle. A full fused tetanus, those frequencies are coming so quickly that we go right up to maximum contraction. We're at the greatest that muscle can do, and we're never going to see it go beyond that point with the number of stimuli that we have there. Even if we increase the rate and start to put extras in between here, we're still not going to be able to get anything stronger than that contraction that we have at that point. So in the lab, we again start to look at the ability of the muscle to contract, and we have electromyography to look at. And with electromyography, we have an EMG, similar to that what you find in an ECG, say in the hospital setting when you're looking at the heart. It's simply monitoring the electrical activity of a muscle. In this case, we happen to be looking for the electrical activity in the biceps and triceps that is there. And so that allows us to look at that and see when somebody is contracting their muscles as they're holding, in this case, they'll be holding a bag, and we're going to continue to put more and more weight into the bag over time. And we see it in a graph such as this. And so our first graph here up at the top, this is somewhat similar to the LT website, except that this is the LT simplifies it a little bit, but this is what we would see in the lab itself. And the first one here, the biceps, this is kind of an averaged biceps contraction, kind of smooths things out, makes it look a little bit nicer as it goes, versus the biceps down here in green. This one here is all over the place. This is kind of the raw data. This is the information as it's coming right from the muscle itself. And so it's very up and down as that muscle is contracting. But we can definitely see that the width here is increasing as we add more force to it, versus the other one here just kind of shows us a nice steady incline of the information. The interesting thing here as well is that for the triceps, we have, once again, an averaged triceps. And we can see that the triceps is just slightly kind of increasing in its intensity that's there, and the amount of contraction that is occurring. And again, when we look at the raw data, that raw data is slightly becoming wider as we added more weight into the bag itself. And this, what's happening here is, if you remember back to anatomy, we talked about antagonists and agonists to muscles and synergists and things like that. And with those muscles, we definitely do have antagonistic muscles here between the biceps and the triceps. But at this point, the contraction of the triceps is not trying to override that of the biceps. It's actually there for stabilization of the joint. And so we're trying to stabilize the elbow joint so that it stays in one place. Nothing slips or slides or rolls or anything in that particular area itself. And so this allows us to see that contraction of both sets of muscles as they're working in real time. And from there, we'll take another look into the muscles the next time around.